Okay, welcome back everyone to the final leg of this meeting. And to kick us back off with the final talk, I welcome Abimbola to chair that talk.
Thank you so much, Oliver. Thank you. So, Jacob, can you share the screen? Okay, thank you. Good afternoon, everyone. I would like to appreciate the organizers for putting this workshop together for us to come together, all the PDRAs under the Faces of Life grant, and to showcase and also for possible networks and also for possible collaboration. My name is Abimbola Olulano from the University of Sheffield. And today I have with me Marco Maury from the University of Edinburgh and also Jacob Biboy from the University of Newcastle. Next slide, please. So our project focused on looking at and trying to understand the physics that underpins antimicrobial resistance. So in a simple term, we are trying to understand why bacteria resist or is also resistant to antibiotics. So this is already a well-known uprising topic in the literature and also in the news, in the fact that if care is not taken, the next pandemic could be antimicrobial resistance, which are also known as superbugs. In fact, on WHO website, it's already stated that by the year 2050, we could have about 10 million deaths from antimicrobial resistance, which is even much more going to be higher than that of cancer. So to be able to focus on this, we actually focus on two major pathogens. The first one is Staphylococcus, which is a spherical cell, and next one is also E. coli, which is rod, and that's what you can see on the screen. And I would also want to say that to be able to tackle this problem and understand the physics behind antimicrobial resistance, we need to have a multidisciplinary approach, and in such a way be able to have collective of expertise from different backgrounds, and that starts from single molecule optics to using bioengineering and also biochemistry approach to studying the E. coli and Staphylococcus, and down to use the atomic force microscope to look at the molecular organization that are associated with the cell wall of these pathogens, and at the same time trying to also use molecular dynamics to study and to be able to have mechanical properties that are associated with the cell wall of these pathogens. And we have four main universities that host this project. We have University of Newcastle, we have University of Cambridge, we have University of Sheffield, and University of Edinburgh. And for today's topic, because of time, we will focus and talk only on the E. coli part of the work, And at this point, I'm going to hand over to my colleague, Marco Maury, and he's going to talk about the biophysics of the cell wall of E. coli. Over to you, Marco.
Thank you, Abby. So as Abby has already said, we are going to focus only on the Escherichia coli. And that's a bug, as you see in the top left picture, that is an average width of half a micron and a length of 2 microns. So we are going to focus on the cell envelope, that is the one that is represented at the center of the picture. And from bottom to top, the cell envelope is made by an inner membrane that is in contact with the cytoplasm of the cell and has to sustain a turgor pressure of 2 atmosphere. A central part that is called the periplasm that lodges a peptidoglycan cell wall that for short we are going to call PG, so stands for peptidoglycan. And this part is actually the one that protects the cell from lysis and that confer the shape. And finally, we have the outer part that is called the outer membrane that is in contact with the environment, the outside of the bacterium. So actually, peptidoglycan is important. And for that reason is also suitable for being targeted by several antibiotics. Unfortunately, in some cases, bacteria are able to develop resistance to this antibiotic. And in this case, we are going to chat about antimicrobial resistance. So the cell actually grows and during growth needs to enlarge the PG succulus and insert new material in the old existing layer. This is a complex mechanism, so here is just an exemplification how it works very quickly. From bottom to top in the cytoplasm, the peptidoglycan precursors are synthesized. Then they are flipped through the inner membrane and put together and arranged in the old part of the succulus. So this part is done by several enzymes. So for example, the transpeptidases, TPases that link the peptides that are the one represented in as dots in the picture. Then there are other enzymes that are the glycosyl transferases, GTases, that are able to link the sugar backbones. So the sugar backbones are the one in gray and white. And finally, there are also hydrolases that are able to cleave all the links. So this one basically is the mechanism by which the cell replaces the old part of the succulus with the new material, and needs to be strictly regulated by different enzymes that we've already seen. And there are different enzymes that are working in during the elongation and the division processes. So next slide. All of these leads to a arrangement of the PG succulus that in the classical view is seen as order arrangement, as you see in the central picture, where there are mesh of glycans that are the one represented by polygons, that are the backbones, and connected by some short linkers that are the peptides, again in as dots in the picture. And finally, we have these linkers and glycans that create a network that has some holes, some pores, that has the size of 10 nanometers, and the final network has a thickness of 2 nanometers on average. So we need to link the molecular information of the PG layer to the cellular scale, and to do that we need two different approaches. First of all, we need to measure quantities by physical quantities. And also, we would like to understand how they're organized. To do that, we are also developing theoretical modeling. The main aim of our consortium is to find out the weakness that can be exploited to target these resistant bacteria. And this obviously needs a multidisciplinary joint effort. Since the PG succulus is so complex, in the next slide, Jacob will tell you how this succulus is assembled and the composition of the succulus. So please to you, Jacob.
Thank you, Marco. The peptidoglycan is basically composed of glycan chains of N-acetylmuramic acid and N-acetylglucosamine. The pentapeptide is a short peptide of 5 peptides that are linked by crosslinks. And in bacteria, the peptide is in the peptidoglycan, you will find peptide length of from 2 to 5 peptides. And as the bacteria grows, the 5 peptides can be trimmed down to 2 by PG hydrolases. And this linking of glycan strands is by transglycosylases, and the linking of the crosslinks between 2 peptide strands are by transpeptidases. The normal linking process is between the fourth amino acid of one peptide and the third amino acid of the next chain. But as it matures, sometimes we find links that are between the third peptide of one and the third peptide of the other. So a combination of linkages and length of the amino acid chain gives the shape to the molecule, to the bacteria. And this, the PG we digest with an enzyme called muramidase, and we study it by the composition of the peptidoglycan by using HPLC. And this gives us information of the crosslinks, the average glycan length in terms of the number of glycan molecules there, the maturation of the peptide, and any modifications that can be seen on the peptidoglycan. And the formation of peptidoglycan is a multi-protein process, and the peptidoglycan assembly is or biogenesis is by two different systems. One that works on the lateral cell wall, and that is called the elongosome, and the next is at the divisome, and at the division site, and it's called the divisome. The elongosome and divisome require major synthesis, PP1A and 1B, for the function of the elongosome or the divisome complex, and also have a very important transpeptidase of each of these biogenesis systems have a particular transpeptidase that is needed for its activity. In elongosome, it's PP2, and in the divisome, it is PP3. If you perturb the elongosome, the divisome can take over, and the bacteria will grow as small filaments for a few generations and ultimately lyse. Similarly, if the divisome is perturbed, the elongosome can take over, and the bacteria will grow as spheres for a few generations and ultimately again lyse. So for our work, we have chosen mesalamine as a target, as an antibiotic of choice for study, because mesalamine is specific only to the PP2, the transpeptidase in the elongosome complex. And also it is active against gram-negative bacteria. Other beta-lactams have a problem that they affect multiple synthase enzymes like 1A, 1B, and multiple targets. But mesalamine is targeting only PP2 of E. coli. Again, mesalamine is also used for treatment of urinary tract infections and is clinically relevant. And once PP2 is inactivated, the bacteria turns into a sphere, divides for a few generations, and will ultimately lyse. These work in bacterial biology, we can develop strains that are more resistant to antibiotics, and mesalamine resistant or increased resistance of mesalamine in laboratory strains can be achieved by two ways: by increasing the level of PPGPP, secondary messenger system, and this increase in PPGPP causes the transcription and translation of multiple proteins in the cell, and also increases the expression of FTSZ that is responsible for the divisome assembly. Increase in FTSZ is seen to relieve the pressure of the elongosome, and the cells can grow as spheres or divide as spheres. And we can increase FTSZ expression using a plasmid also, and that's how they found out. And one important thing is that the strain always needs the PP1B, LPOB, that is the major synthase in the divisome complex. And these are the requirements in the laboratory, we can do it, but recently, what Tulin and Anderson, clinical biologists, found was a strain that was deleted for cis-B, regulator of cysteine biosynthesis. And they found that this strain was a stable resistant strain, and the resistance was much higher than in the laboratory strains. And deletion of this was not down, deletion of the gene didn't cause resistance due to the increase in PPGPP. That was another finding of theirs. But they found that there was a very, about 30% increase in the FTSZ levels, a 60% increase in the PP1B and its activity LPOB. But other than this, they couldn't find a reason why this strain is resistant to mesalamine. And that is why we use this strain and mesalamine to study the biophysical and the biochemical mechanisms of mesalamine resistance. That's the talk for now. And from now, I'm going to hand over to Marco to talk on some preliminary work on the reasons for this resistance.
Thank you, Jacob. So actually, I'm going to present a work done by a former member of our lab. Next slide, please. Rebecca Browers. So we have already seen that meselinam is able to inhibit the elongation, but not the division. So maybe we can focus directly on dividing cell and understanding some condition for obtaining such division in the presence of meselinam. So as you see in the picture, so we exemplified dividing cell in the presence of meselinam as a sphere because they get spherical with antibiotic, and they have a specific volume and also a surface. So we also found out a parameter that can tell us when a successful division happens, that is H, basically the division between the two cell walls of the dividing cells. So since meselinam is able to inhibit elongation, but not division, so cells are still going to grow and divide, we can express the increase of volume normally as an exponential growth law, as you see from the first equation exactly. And we can also suppose that the surface increases at a fixed rate y, as expressed by the second equation. So from the picture, we can also compute out the volume and the surface as expressed by the two equations at the bottom of the picture exactly. And the successful division, as I told you, depends on this parameter H that should decrease over time. So when the division is successful, when actually the surface synthesis is able to overcome the volume synthesis. So if you produce much more volume than surface, actually the cell lyses. If the surface is enough to keep all the volume inside, the division is successful. So once you plug the volume and the surface in equation 1, 2, and use also the constraint 3, you obtain for a successful division that there is a fatness threshold, so basically a radius that tells you that the division can be successful. And this fatness threshold is expressed by the equation on the bottom and depends on the rate of our synthesis over the volume expansion. So equipped with Vision, we also tested the prediction of a model. So next slide, please. So basically, the model tells you that there is a radius for which the cell undergoes lysis if the cell grows much larger than this radius. And we call it fatness threshold. So how we tested the model, we made grow the Escherichia coli into different media, a rich medium and a poor medium. So that has a different growth rate. And in the rich medium, you can read the plot from left to right. So basically, the cells start growing happily as a spherocylinder, then the meselinam is put in the medium and the cell gets spherical, but can actually grow a little bit more and divide as a sphere, but exceed the fatness threshold, that is the one indicated in red. And at this point, exceeding the fatness threshold lyses. While instead in a poor medium, the cell is not able to exceed the fatness threshold, so gets still spherical, but divides continuously as a sphere without lysing. So that one actually was a test for the model, and we can say that. Oh yes, please, thank you. And we can say that we are able actually to catch a complex medium-dependent effect of this meselinam on the cell wall by simply setting up a very simple mathematical model. And the key ingredients are the balance between the growth and the surface area synthesis plus geometric constraint. So we also tested further this model. Next slide, please. So Leonardo Mancini actually did an experiment in a microfluidic chamber where bacteria are arranged in a line inside this chamber, and they are able to divide. And we provide both meselinam and nutrients to the bacteria. And you will see in the video that while we provide meselinam to the bacterium, So you will see that meselinum kicks in the first video. They're getting larger, and at a certain point, they are going to get spherical and explode. Here it is. So sphere and then explode. In the second video, instead, you see that they are going to grow without exploding. Why that? Because they were put in a chamber that was narrower, so smaller, so that they cannot exceed the fatness threshold, and these geometric constraints, or these mechanical constraints, enable them to still divide without lysine. So that one was again another confirmation of the goodness of our model. And since we are interested actually in understanding the PG succulus, the lichen, in the following slides, Jacob is going to tell you how we extract and purify these succulus. So please, Jacob, is up to you.
Okay. The peptidoglycan purification step is quite simple. We just collect grow cells in media and then harvest them by centrifugation and then boil in SDS to inactivate the autolysins and also to solubilize all cellular components other than the peptidoglycan. And once they are boiled, and then the peptidoglycan is washed free of SDS. And then we do an enzymatic digestion to remove any peptides that are covalently bound to the peptidoglycan, or glycogen moieties bound to peptidoglycan, and then again boil in SDS to remove the enzymes that we just added, and then harvest them and wash free of SDS. And this is then either used for atomic force microscopy, on which Abby will talk about soon, or we use it for HPLC analysis. And as I said, as I alluded before, we use an enzyme called miramidase to break up the peptidoglycan cyclase, and use an HPLC to see the profile of the PG profile of bacteria. And in our case, when we studied the peptidoglycan profile of sensitive strain, that is a wild type, and the meselinum resistance B strain, we found that in normal conditions, both the peptidoglycan looks exactly the same. There's no difference in the composition of the peptidoglycan. And when we treat this with meselinum, again, what happens is that the monomers reduces. And monomers are basically single glycan moieties with the peptides attached, and multimers are where they are cross-linked to each other. And in the monomer content reduces while the multimeric cross-link content increases in the sensitive and the resistant strains. And we do not see any difference. And also what we note is that there is a decrease in the length of the glycan chains, as we'll see in this, the numbers, the values that we can extrapolate from the chromatogram. And as you see, there's a reduction in the monomers in both the strains when meselinum is added, the increase in the trimers or the complexes that are formed, and the linkage is also increased. And that average, the chain length is the number of glycan moieties between crosslinks are reduced. Other than this, there's overall, there is no difference between sensitive and a resistant meselinum resistant strain. That with that, we thought we would need, we would understand better if we can find any information using AFM to know there's any difference between a sensitive and a resistant strain. So I will hand over to Abby, who will talk on the AFM part of the work. Thanks
so much, Jacob. So as Jacob have said, usually he has to send over the succulate to me, but the most important thing is that we want to use atomic force microscope to actually scratch the surface to see how this PG looks like. And in a basic term, how does the AFM works? So the AFM is a surface profiler that uses a very sharp tip, like a cantilever tip, to feel the surface and profile the surface, and then give it the topograph of the surface. And how it does it is that between the tip and the sample of interest, and this sample could be any kind of sample, be it a conducting or not conducting sample, or just biopolymers or any biological sample. So the most important thing is that this force between the tip and the sample causes the cantilever to bend as it raster across the across the surface. And this is being monitored by the laser spot that then generates a reflected beams into the photodetector that serve as an input for a feedback loop. And here is an example of the kind of mode that we used, because you can actually use different kinds of mode for in atomic force microscope to operate the AFM. So you can use the contact mode, you can use the peak force mode, but for the E. coli, we actually used the tapping mode, in which we have to oscillate the cantilever near its resonance frequency as it scans across the surface. Okay, so now you can see how the tip oscillates and gets closer to the surface, having a reduction in the amplitude of oscillation and scan through, and then that gives you the Z topograph. Next, please. So as I said, that AFM is a surface profiler. So you cannot just have the tip in the solution and have the sample only in solution. So it's not a solution-based kind of technique. It's a surface-based technique. So you need to immobilize your sample on the surface. So in our case, we're using a mica sheet. So as soon as I have the circulate from Jacob, I have to immobilize this on a mica sheet. But because the cell envelope is negatively charged, so I need a bridge. So the first thing is to actually passivate the surface using polyhedrontine, and afterwards immobilize the circulate based on the dilution or based on the concentration, and that lasts for 10 minutes. Then wash sporously, like just pipetting up and down 3 times to remove whatever excess material that I have that are not physiosorbed, just remove them, and then image in trace buffer, which is just 10 millimolar trace using, this is actually a typical picture of the AFM that we have in lab, which is FastScan. Next, please. So as Jacob explained earlier on, so these are actually a rod-shaped bacteria. So it's even, for the wild type or for the mutant, which is the CISB that is resistant to meselinum, both are actually rod-shaped. But interestingly, as he mentioned, when you treat such with meselinum, what you have is actually a rounded spherical bacteria. And I will also mention that even for CISB, although you are not seeing the micrograph, we still have the spherical shape once it is treated with meselinum. In addition to this, we step further because we just do not want to just have a qualitative images alone, but we want to further quantify. So we quantify the width of this rod, and if you look at the result on the right-hand side, you would see that in terms of the width of the rods, you don't basically see any difference. They fall on the same range between 1 to 1.5 micron in terms of the width, which is the center of mass of the rod. And again, in terms of the length, which is total length of the rod, you also see that for the wild type and the CISB, they also fall in the same range between 1.5 to 4 micron in terms of length. But the main difference is actually when they become spherical. In this case, we are assuming that we have a full spherical and we're using the diameter. And you can clearly see that in that case, with respect to either width or the length, if it's with respect to width, we actually have like 4-fold increase in terms of the width of the sphere. And if it's with length, we have about 2-fold increase. Next. So beyond just looking at the shape and quantifying the width and quantifying the length of this bacteria, the most important thing is actually, since we're looking at the cell envelope, we want to see the molecular organization of this cell envelope. We want to see the glycans that Jacob and Marco have spoken about. And in that case, the kind of AFM we use actually allowed us to see high resolution images. And these are typical images that you're seeing. So we are first of all looking at the sidewall, not at the post, just looking at the sidewall and seeing the kind of organization we can see. And as you can see, you can clearly see we have glycans that are actually spread across, they revolve around the cell wall. So, and you can clearly see that they are fully oriented to a given direction with respect to the plane of imaging. And at the same time, if you look at it across the longitudinal, you would see that they are not perpendicular, but they are fully oriented towards a given direction. Then when we saw this, we said, okay, because the width of these glycans that you can see, they are close to between 1 and 2 nanometer in terms of width. So wanted to also see whether we have the same representation for the CSB, which is also the resistant strain. Next, please. And when we look at that as well, we also have a very good resolution, and you can also see that it's also well-oriented and an ordered structure. So far with the images that we have, we could not see any major differences, just reflecting what Jacob found from the biochemistry analysis point of view as well. We did not see any difference. The only thing is that they are both oriented with respect to the sidewall. Next. So at this point, I will hand over to Marco, and it's going to talk to you about the mechanical modeling that is working on putting in place to be able to study the mechanical properties that are actually associated with these glycan strands. Over to you, Marco.
Thank you, Abby. Okay, thank you, Jacob. So as we said already, we are also working on modeling, and here I'm going to present a preliminary result of this mechanical model of VPG sacculus. So why we are doing this model, since there are other already model present in the literature? The classical view of VPG sacculus is that is a well-ordered structure, as you see in the first picture, exactly there, where glycans are organized in hoops, as you see in green in the picture, and they are connected by peptide linkers in red. While from the AFM data, as Abby already suggested, we have a different kind of picture, and specifically the organization is not so ordered, but depends both on the position in the cell wall, so if you are in the center, in the cylindrical part, or in the poles, and also in the growth condition. For example, in the picture, you see a bacterium that is happily growing, it's a spherocylinder, and in this case, the glycans are long and well-ordered. While in presence of stresses, the bacterium gets spherical, for example, during mycelium action. And in this case, the glycans are shorter and the order is much reduced. So basically, the older models concentrated themselves in studying the order part. We would like to transit to the disorder part. In the following, you are going to see only the initial part of this job, and I will tell you what's going on. So we are interested in understanding the action of the antibiotics, and also how the antimicrobial resistance is developed in these cases. So next slide, please. So we focus on a very coarse-grained model that is based on Beden springs, and the idea is to mimic the biophysical properties of the sacculus. So the unit of the model is the one represented on the left side. So there are beads in blue that represent the center of mass of glycans and peptides, and they are connected by springs in green, glycans and peptides in red. And once you put together several units, you obtain a patch, yes, the one in the center. And we would like to understand how this patch, that is now geometrical actually, very ordered, is stable or behaves upon damages, also in the presence of antibiotics that can cleave some crosslinks and damage the patch itself. On the other side, we are also working with the same model to understand some AFM data, because the classical view in models that are present in the literature for the AFM analysis focus on an homogeneous and isotropic sample. While in our case, we have seen that the sample is not homogeneous, is not isotropic, and the AFM tip is actually has a size that is very similar to the one that is probing. So basically, the size of the glycan and the peptide. So in that case, the normal models are failing, and we would like to extend it from a continuous to a discrete model. How we do it? So the next slide, please. Yeah. So we developed this model that is basically very simple. So we start again with Newton dynamics. On the top, you see the Newton equation, which sum up all the forces that are acting on the sample. We work in an overdamp situation, so that the equation can be a little bit simplified. And after we discretize it, so that we can implement it also in the computer with an algorithm. So our algorithm is working at time 0. We choose the orientation of the glycans, the cross-link density of the peptides, and also the length of each glycan chain. Then we compute the forces on operating on each bead from the potential energies. And the forces we are considering are spring forces. So the difference, extension between distance of 2 beads. The bending, because glycans are going to be straightened up usually. So we have also thermal fluctuation. So the Brownian motion, basically. And also the turgor pressure, because there is a difference of pressure between the inner part of the cell and the outer part of the cell, and the PG cycle is the one that sustain this pressure, so that the cell do not lyse. And so, as I told you, we discretize the system, and we calculate after for each time step the forces that are acting on each bead and the position of a bead at the time steps after. So we can loop over this chain and obtain the dynamics of the beads over time. So next slide, please. And the simplification of this, yes, please start. Of this idea is that, for example, the mass perturbation of this unit. So I pinch here a bead, and after I remove the pinching end, the system relaxes back to a new equilibrium position. So that is the easiest stuff that you can do with this kind of system, but we can do also much more complex things. And the one is we're going to see in the next slide is a little bit complex. Yes. So in this case, we are analyzing the action of the AFM indentation. So we take again a patch that is well ordered, and we push in the yellow point with an AFM tip. Yes, please. Yep. So you will see the bead that goes on due to the action of the AFM tip. And at a certain point, we remove the AFM tip so that the mesh can relax to a new equilibrium condition. So from that, we can also extract the action of the AFM tip on the mesh and understand the force indentation curve. So that are just two very simple examples, and for an order geometric, very order structure. I'm not going to present you something regarding more complex ideas and disorder structure, because we are still working on them. So it's a preliminary work. But the idea is that we'd like to understand and develop this realistic model to study the mechanical properties of the PG layer. And as I told you, we're working on two different ways. The first one is understanding the effect of antibiotics. For example, in this case, there are uncoordinated enzymes that are chopping peptides linkers. So they are undermining the stability of the succulus. And the other case is, as you have seen in this very simple simulation, to understand the action of the AFM data on a not continuous but very discrete sheet of PG layer. So that one was actually everything for this preliminary part of the model. And with this one, I will give the word to Abby that will conclude and give a summary.
Thanks so much, Marcos. So in summary, thus far from the results shown by Jacob, you've seen that he was able to review to you using the biochemical studies based on HPLC that shows that there is no major difference between the peptidoglycan composition of the wild type and that of the resistant strain. And also from my hand, I've also shown you how we've been able to see that we have oriented glycan strands at the body of the cell wall. And we are trying then to understand what happens for meselinum for the strain when it's not being treated with mesenchymal, when we have rounded spherical structure. Then at the same time, from Marco's end, just for you to have like a kind of take-home message, he was able to show you some simple physical model that is working on, that is developing to be able to see what happens to the mechanical properties of the PG layer, and at the same time to also show, we've shown you the effects of confinement on the cell growth that then stop the cell from lysing once they are in a confinement microfluidic chamber. And at the same time, also we've also shown you how we are trying to also understand what happens when the sacula is exposed to antibiotics or when it's being indented by AFMT. Next. So in terms of the future goals, from the Jacob's point of view, it's going to be looking at the interplay between the PG synthesis and the hydrolysis and the recycling enzyme in terms of the CSB mutant, which is the mesenchymal resistance strain. And at the same time, it's going to be looking at the importance of the PG modifications and also site and the effects of the site modifications and as to how this contributes to the mesenchymal resistance. From my own point of view, so far we've only focused more on the core, on the body of the saculae. So we are now going to be looking at the post and trying to see, do we have short chains? Do we have long chains? Do we have less and disordered structures? Then at the same time, we'll also be looking at the rounded cells that's when the rod is treated with mesenchymal. We want to see whether we have short chains, whether we also have lack of orientation and disordered structures. And from the theoretical part of the work, which is done by Marco. So now he's working on how to do a kind of prediction of the mechanical properties of the PG sacculus and also extend these to the resistance strain, and at the same time trying to work on how resistance actually alter PG synthesis and also the stability of the sacculus. Next. So in terms of the burning question, so the main question that we've seen that speed out of our work thus far is molecular organization. So we are trying to now say, okay, how does molecular organization on the nanoscale controls the function of this cell envelope that we've seen in terms of the details we've seen thus far, and just trying to link architecture or structure to function. And at the same time, we're also trying to also see if there is any relationship between molecular organization and the mechanics of the cell, of the cell, which is actually the main part of work from Marco point of view, trying to see whether when we have ordered and a kind of oriented structure, does that influence the mechanical properties as well. Next. So in terms of the people that are actually working, so we have from Cambridge, we have Pietro and Leonardo. They are the one using the microfluidics chamber to actually study cell division, growth rate of the cell and the likes. And in Newcastle, we have Waterman and Jacob, and they are the one doing the biochemistry analysis of the composition of the PG with respect to different strains as the case may be. And in Edinburgh, we have Roslyn and Marco. Marco, you said you've heard from him. So they are the one working on the mechanical properties, modeling of the work of looking at what are the associated mechanical properties to the PG that we are looking at. Then from my end, we are based in Sheffield. So Hi and Jamie, we are the one working based on the ultrastructure using atomic force microscope on the nanoscale to fully reveal the architecture that is associated with these strains. Nice. So I would like to say thank you so much for listening. And we are so happy to take any question that you may have. And at this point, I also ask Steve to give the poll question for our side. That's the mini questions that we have. So please, attendees and the audience, feel free to answer the questions. Our own question is actually based on true or false, so which is very clear. If you have listened to our talk thus far, you'll be able to answer these questions. So while we are doing that, you just need to click and submit your answers. I would like to look at the Q&A. So if you have any questions at all, you can actually pop it in the Q&A section for the attendees, and for the panelists, you can actually ask the question via the chat window. So here is a question which is directed to Marco, and it says, for modeling AFM data, if you model the indentation, would you add in a yield strength to see if you can get the reverse of the molecular pooling experiment, looking at going beyond the elastic phase and looking at the yield strength? Then the second part is saying, for example, so STAPS in AFM force as a measure of aspects of rupturing of the peptidoglycan network.
Okay, so thank you, Mattis. It's actually an interesting question. We are trying to develop this model as close as possible to the real data that we are catching from the experiment of AFM guys, so from Abby. And yes, the idea is actually that we already have this kind of rupture structure, and also inhomogeneities in the system, and something that goes a little beyond the Hooke's law. So we are catching already the difference between glycans and peptides, while glycans can stretch continuously according to Hooke's laws. Peptides are not going to do that. So the idea is that, yeah, we are able to include this yield strength and rupture specificity of the PG sacculus. But the results are too preliminary to be compared to the experiments, I think.
Thank you. So we're still waiting for questions. Okay, here is another question from the Q&A. So here is a question from Nangao, and says, not really a question, but it might be worth mentioning that some AFMs allow the user to do aqueous imaging. In addition, there are some noise like drift in AFM images shown. So for the images that I actually showed, they were all captured in aqueous solution, in buffer solution. So they were all captured in the liquid. So these are images obtained in the liquid, but not just that, but we have to immobilize the sample on surface. Yes, there are also some issue. It's possible that you could have drifts, which is it's possible that you have such things happening, but depending in terms of the how stable your system is and the kind of tip you're using, that you can control to some extent. But for the images that I have, there is no a typical or well-pronounced drift, because they are well fixated and well, they are very well on surface. So we could be able to actually obtain a nice images as I've shown. And okay, so no. So now, no, so these are actually, so in this experiment, because of the structure that we have for the E. coli, so you have the outer membrane, and then in between you have the sacculus. So it's not on live cells. These are actually dead cells. These are cells that has been boiled so that we can be able to extract the cell wall out. So these are extracted cell wall immobilized on surface. So it's not on cells. It's not on living cells. These are actually dead cells that we already have. So we're only focusing on the peptidoglycan that has been extracted from the cell, immobilized on surface. So it's flat on surface on a single sheet. And on that, we are just looking at that single layer and seeing the way glycans are. Okay. So she said, flow cells for AFM aqueous imaging. Can you explain further, Nangao? What do you mean by this? So if we have to, because we are focusing on this part of this, one could use it that, yes, it is possible. It is possible for you to have, okay, you mean a flow. So what you're saying, you're actually talking about a flow cell, like a liquid cell, yeah. So a liquid cell to be able to have an inflow and outflow. That you can do. And if you are doing that, it's going to be useful, for example, if you are looking at an immobilized cell. So for example, if you have, for example, in the case of Staphylococcus that we do immobilize that within grids. So you can actually put that, you can have that on a grid that you've already immobilized on a live cell, you immobilize it on the grid, and you have an inflow of liquid that goes in and out while you're imaging. That you can do. But for the case of already a dead cell where you just only have extracted cell wall, there's no living cells, extracted cell wall. Yes, you can use flow cell just to keep the buffer. That's the only thing, but you are not seeing any dynamics that is related to the living because it's not alive, it's a dead cell. Does that answer your questions? Okay, great. So then we have another question. Okay, I think this is fine. And let me just, so we could continue on the chat window for many more questions. So at this point, I will just give an answer to the questions that we have. So the question number 1, the answer is true. Number 2, the answer is true. Number 3, the answer is true. 4, the answer is true. 5, the answer is false. 6, the answer is true. And also the last question, the answer is false. Thank you so much. So at this point, I'm going to hand over back to Oliver, and he's going to take us through the next thing. Thank you.